We take blood from a person and we extract all of the nucleic acid from it, particularly the nucleic acid that would be like RNA or DNA, as you may have heard of it.
This isn't a scene from the 1978 movie that was filmed in these hallways.
Invasion of the body snatchers.
Dr. Pandori, interim director at the Public Health Lab in San Francisco, is talking about innovative testing technology that's keeping his lab on the cutting edge.
And we have a special, highly specific test that can amplify even tiny amounts of that material and detect it. So if there's HIV present in the blood, as it might be if somebody was perhaps recently infected with HIV, we would be able to detect those minute amounts and use that as a diagnostic for HIV infection. In the lab, it's actually hard work because we have to test a lot of specimens using a method that we had to adapt and adopt to this particular application. But in the wider field of medicine and in public health, I think it's been really quite impactful. Current antibody tests aren't really perfected for catching people during that infectious period. This test can catch them, so it allows us to do better public health work because we're not only finding the most infectious individuals, but we're finding them at a time that our investigations are most fruitful because they can remember partners and communicate more clearly with the disease control investigators.
Take these out and see what's going on. This is a pure culture of Neisseria gonorrhea.
The lab is also advancing efforts against drug resistance in gonorrhea.
Because fewer and fewer antibiotics are being developed, we're really being backed into a corner with regard to our treatment of gonorrhea. So we're starting to realize that we're going to need other weapons, so to speak, against this infection. And one of those is a test that was actually invented in this laboratory several years ago, which was a test that based on the detection of genetic information in gonorrhea, which allowed us to make a very rapid assessment of whether or not a particular class of drugs would function against a particular infection that a person harbored. So that at the time of diagnosis, we would already know whether or not a drug worked, and we wouldn't have to take the risk of giving a drug and hoping it would work.
The work that we're talking about with the gonorrhea study is just a part of the overall work in looking at molecular methods in the laboratory and how they can be used to improve health and public health.
San Francisco's lab is one of 3 sites in the U.S. that will begin a clinical trial later this year to assess the viability of this test.
It's exciting in that this laboratory is constantly pushing forward the boundaries of diagnostics, and particularly for STDs like gonorrhea and also for HIV. And the reason that we think that it's so important and why I am so proud to be part of the branch that this laboratory exists in is because that type of innovation is really what improves public health for the people of San Francisco.
I think our greatest strength with regard to infectious disease is technology. I think by appreciating this technology, we will move into a phase where we're not only detecting organisms, but we're simultaneously describing them, because that's really what ultimately is most important in the context of understanding organisms and how they evolved. Is it's not just whether it's present or not, but how dangerous it is and how much it's changed. By being able to assess the genetics in a more complete way and in a very rapid way, we'll be able to detect and describe organisms in what you might call real time, and that's how I think we stay a step ahead.